Hi, folks. Welcome to the fourth lesson of Unit 2 of the Higher Human Biology course. This lesson is on ante and postnatal screening. So, we're going to kick off with the antenatal screening to start with. So, antenatal screening is a process which identifies the risk of a disorder, the risk of a disorder. And once they've established the risk of a disorder, they can then undergo further tests, and what's called a prenatal diagnosis can then be offered to that parent. There's a variety of techniques that can be used to monitor the health of the mother, the developing fetus, and the baby. So all three will be monitored at various stages of the pregnancy, and then obviously post birth as well, postnatal. So the first thing that happens in terms of assessing the health of the mother and the fetus is something called an ultrasound. So we've got ultrasound imaging here. Now, pregnant women are given two ultrasound scans. The first one's called an anomaly scan, and that detects any serious physical abnormalities in the fetus. That's the big one where they're looking for anything they can see on the scan, and that's usually done between 18 and 20 weeks. The dating scan is one where they use it just to establish the stage of pregnancy that the mum's at and the due date. And what that also helps them with, if they know the pregnancy stage and the due date, that will then be able to be used in conjunction with tests that look for marker chemicals. That's the chemicals which increase and decrease in concentration during different stages of pregnancy. And they will vary throughout. And this dating scan is done between 8 and 14 weeks. From my own experience, they're usually done at 12 and 20 weeks. So I always say to my students, just if you only want to remember two numbers, remember 12 and 20. But the SQA give you a range here of 18 to 20 for the anomaly scan and 8 to 14 for the dating scan. And you can see a couple of images here showing you what an ultrasound scan looks like and what the procedure itself looks like up here as well. So throughout pregnancy, from the moment the mum realises that she's pregnant and throughout the rest of the pregnancy, there'll be routine blood and urine tests. Now, they're carried out throughout the whole of the pregnancy to monitor concentrations of things called marker chemicals. So there's a few examples here. We've got HCG, which is human chronic gonadotrophin, and they'll monitor those levels in case of abnormally high levels at any point in the pregnancy, because high levels of HCG may indicate Down syndrome. Another one they look for is one called APL, and again, that can indicate another condition called spina bifida. And the last one is looking at protein that is called a Papa protein. And they look at those Papa levels and they may indicate Down syndrome as well if those Papa levels are particularly low. However, caution must be had when you're measuring these marker chemicals throughout pregnancy. And that's because if you measure a chemical at the wrong time, then it could lead to something known as a false positive result, which is where you believe that there's a high HCG level, for example, at a certain time of pregnancy, and you think that that indicates Down syndrome, but actually, if you get the date wrong, the due date, and therefore the pregnancy date wrong, then that can obviously mean that you're actually measuring the chemical at the wrong time, and so we get a false positive result. When we do get a atypical chemical concentration, so something that we don't expect, like the high HCG, high APL, or low PAPA, if we get one of those, that can lead to something known as a diagnostic test taking place. And a diagnostic test is a test to diagnose formally the condition that the developing fetus may well have, to see if they've got a medical condition. And there are a number of different diagnostic tests that they use. The two that you need to know about for your exam are amniocentesis. And that is one where they place a needle into the amniotic fluid. That's the fluid that surrounds the developing fetus. And they extract some of that amniotic fluid, which will contain some of the fetus's cells. And what they can do with those cells is they can then culture them in a lab and use them to investigate any chromosomal abnormalities a bit later on. But they diagnose these potential congenital abnormalities by examining the cells that are found in the amniotic fluid, okay? So that's amniocentesis. Chorionic villus sampling, or CVS as it's more commonly written down, is used to diagnose these potential congenital abnormalities by examining cells, this time from the chorion in the placenta. Okay, so this is the chorion here. Okay, that's the area there where they'll extract the cells from. And I've actually seen it in past papers where you're asked to indicate on a diagram where those cells would be extracted from. Okay, so it's from the chorion area here on the placenta. Now, CVS is useful because the main advantage is it can be carried out earlier in the pregnancy than amniocentesis can. However, the downside of CVS is it carries a higher risk of miscarriage, okay? And again, this is a really common question in the exam where they'll say, what is the advantage of amnio and the disadvantage? So the advantage for amnio would be, it's got lower risk of miscarriage, but the disadvantage is it's carried out later in pregnancy, and it's vice versa for CVS. And the other type of question is where you're asked to compare them. And again, you'd say CVS can be carried out earlier, whereas amnio is carried out later, etc. Okay, so knowing the various advantages and disadvantages of both CVS and amniocentesis will help you for your exam. So the cells that we obtain from either the chorion or the amniotic fluid can then be cultured, okay? And that simply means they're grown within a dish usually. Okay, we multiply the number of cells that we have. And once we have sufficient numbers of cells, we can then use them to produce something known as a karyotype. And that will help us to diagnose a range of various chromosomal conditions. Now, a karyotype shows all of an individual's chromosomes arranged as homologous pairs. And we can see some karyotypes here, okay? We can see the 46 chromosomes arranged as 23 homologous pairs here, okay? And we'll note that each chromosome in each pair is of the same size and approximate shape and so on as the pair that it goes with. And you can see if you look at different pairs, you'll see slightly different sizes and shapes for those chromosomes as well. Now, when they look at these chromosomes, they can see, for example, you can see an example of Down syndrome here, which is it's called trisomy 21, which is basically where they've got an extra copy of chromosome 21. Instead of having a homologous pair, they've got a homologous triplet there. You've got Patau syndrome there, which is trisomy 13. You can see an extra chromosome 13, and Edwards syndrome, which is trisomy 18, which means you've got an extra copy of chromosome 18 there. Okay, so you can see the various chromosomal abnormalities there, and that helps them to diagnose if there's any conditions that can be attributed to the chromosome number. Now, when parents decide to proceed with these tests, there is an element of risk involved, and that needs to be assessed for each individual parent. Okay, there's not zero risk with either of these procedures, although one obviously carries a higher risk with the CVS than the amnio. And obviously, the decisions that individuals will have to make are obviously down to the individual. You know, there's no right or wrong thing to do in that scenario. Okay, and if obviously they get a positive test in terms of a chromosomal abnormality, then they have again another decision to make at that point in terms of continuing with the pregnancy or not. And all of this is obviously quite a difficult time for the parents, and we obviously as scientists respect decisions that are made in that scenario. There are people involved. There are people like genetic counsellors, for example, that you can speak to if you are pregnant and in that situation, and they can help you make the best decision for you. So let's skip ahead to postnatal screening. So this is after the birth of the baby. Once it's been born, we look out for, there's a number of things they test for in postnatal screening, but the one specifically you need to know about for your SQA exam is the test for PKU, or phenylketonuria as it's known. Now, that's a metabolic disorder, which means it affects a metabolic pathway. Now, the pathway it affects is the breakdown of phenylalanine to tyrosine, okay? And that pathway then continues on and eventually will produce melanin. Now, if that enzyme, which breaks down phenylalanine into tyrosine, is mutated in some way, in PKU, it's a substitution mutation, If that is mutated in some way, it will result in it being non-functional. Okay. Now, as a result, the phenylalanine cannot be broken down into tyrosine, and people with PKU will have high levels of phenylalanine if they continue on a normal diet. So, what they do instead is they get placed onto a restricted diet with foods that are low in phenylalanine. Okay, they contain very little phenylalanine. And because of that, we won't have this buildup of phenylalanine, we won't have these high levels, and instead we'll get our dietary protein via tyrosine directly. Okay, so the tyrosine they'll get from their diet in some way by having an altered or restricted diet compared to obviously someone who doesn't have PKU. Now, the last bit of topic 4 talks about pedigree charts. Now, you have already done a little bit about pedigree charts in National 5 biology. And what we have to be able to do at higher is we have to be able to identify and analyze different patterns of inheritance within these pedigree charts. Now, if you are involved in genetic screening or genetic counselling, which I mentioned a couple of minutes before, has an important role when diagnostic decisions are having to be made, you will be looking at those patterns of inheritance, possibly even before you start doing the invasive tests, the amniocentesis and CVS. And what you'll be doing is you'll be looking for any patterns of inheritance which might indicate the likelihood of a genetic disorder being inherited by the offspring. Now, the 4 patterns of inheritance that we need to know about for the exam are what are called autosomal recessive conditions. Okay, now those are typical in terms of the ones we've maybe looked at in Nat 5. A lot of those were autosomal recessive conditions. So, although that might be a new word for you, autosomal recessive, it's one that is probably got a lot of information that you already know about. Okay, you probably already know how they work, but we'll go over it in a wee minute anyway. We've got autosomal dominant. Now, autosomes are chromosomes 1 to 22, okay, or chromosome pair 1 to 22. So the first 22 pairs, or 44 individual chromosomes, are called the autosomes. And you can actually be asked about this in the exam. What is an autosome, okay? So an autosome is chromosomes 1 to 22, or chromosome pair 1 to 22, and you would indicate that it's not the sex chromosomes, okay? Sex chromosomes are pair 23, and I'll just write that down here next to sex-linked inheritance 23. Okay, pair 23 are the sex chromosomes. So 1 to 22 means that the mutation or the genetic disorder is on chromosomes 1 to 22. Okay, pair 1 to 22. A dominant one means that you only have to inherit one copy of the allele in order to get the condition. With a recessive, we need to inherit 2 copies in order to get the condition. Now, there's a new type of inheritance that we've not come across before in higher, and it's this one here, incomplete dominance. And that is where one of the alleles is not completely dominant over the other one. It has some dominance, but it's not completely dominant. And what that means is it's not a binary outcome. You don't either have the condition or not. There's also what's called an intermediate state, which has a reduced expression of the condition. And we'll talk more about that in a moment. And the last kind of inheritance is these sex-linked single gene disorders, okay? And they are recessive ones as well, okay? We need to know about them and be able to work them out for our exam, okay? Now, we'll start off with autosomal recessive because that's the one you're probably most familiar with. An example of an autosomal recessive disorder is albinism, and that's caused by a defective recessive autosomal allele. Now, what does that mean? Well, you need to know about certain patterns that can be shown within a pedigree chart related to each of our patterns of inheritance. And for autosomal recessive, some of the things we should be looking out for within our pedigree chart are the following: that it's expressed relatively rarely, okay? If the condition is expressed relatively rarely, it is likely to be a recessive condition, okay? That's something to look out for when you're looking at the pedigree chart. That's your first clue that it might be recessive. It usually skips generations. So if you have a generation with the condition here, like we can see in this P generation at the top, you can see that it's not present in the F1 generation there. However, it is present in the F2 generation. So that's another hint that it's potentially autosomal recessive. It affects males and females in equal numbers, and we'll establish why that's important later on when we look at sex-linked inheritance. But for autosomal conditions, males and females affected equally. It requires the affected individual to be homozygous recessive or to have two small letters, okay? So anyone with the condition has two small letters. Now, if we look at this example here, these people that are shaded in have the condition, so we know that their alleles, their genotype, is going to be too small a, too small a. And we can fill in everyone who has the condition with that as their genotype, okay? We know that for sure because it's a recessive condition. You have to have two recessive alleles. Now, there are also people that are known as carriers, okay? So, recessive alleles can be carried by individuals who are heterozygous. And if we remember from Nat5, heterozygous simply means you've got a dominant and a recessive allele, okay? Two different alleles. It can result from two unaffected parents who are heterozygous carriers, okay? So that's a small a, capital A, and a small a, capital A there as well, okay? So we've got two unaffected parents. They can still have offspring which have the condition. And we can see that here, okay? We can see that that parent there must be capital A, small a. They must be heterozygote, and so must this parent because they don't have the condition. However, they have offspring that do have the condition. So that gives that A to that offspring there, and that A goes to the offspring there, okay? But they don't have the condition, which is why they've got a capital, okay? So that's autosomal recessive. I think out of the four, that's the easiest one to understand because it's very similar to what we've done previously at Nat5. Now let's have a little look at an example question just to make sure we do understand this fully. So we have cystic fibrosis as a genetic condition caused by an allele that is not sex-linked. A child is born with cystic fibrosis despite neither parent having the condition. The parents are going to have a second child. What's the percentage chance this child will have cystic fibrosis? Okay. So it's caused by an allele that's not sex-linked, okay? A child is born and neither parent has the condition. Okay. So the child, let's call it AA, okay, has the condition. The parents, if they don't have the condition, must be heterozygotes. Okay, we know that. So as a result, what are the four offspring that are capable from these two parents? Well, we can do a quick Punnett square, and our outcome would be as follows. We'd get one homozygous dominant. We would get two heterozygotes, and we would get one homozygous recessive offspring. Okay, so we'd get 1, 2, 3, 4 offspring. And of those four, how many would have cystic fibrosis? It's going to just be the one there, isn't it? Okay, so it's going to be a 25% chance. All right. So that's how we work out an example of a recessive autosomal condition. So autosomal dominant is, again, is similar to what we've done before, but because it's dominant, we only need one allele in order to have the condition. Because it's autosomal, it affects males and females in equal numbers. An example of this is Huntington's disease, which actually I just saw that they've come out with a new therapy for, so that can help delay the symptoms of the condition for those who have it, which is great news. But it affects males and females in equal numbers. It means anybody affected will have an affected parent, okay? So if you're affected, you can see in this pedigree chart, everyone who's affected has got an affected parent, okay? Every single person. So therefore, that is a nice pattern to look out for when you're trying to identify what pattern of inheritance there is in a pedigree chart. If there's an affected parent, then the offspring have it, and we can see that for every generation, we know it's likely to be a dominant trait. When it doesn't appear in any generation of the pedigree chart, however, that will mean it's gone forever. Okay, so if there's a set of offspring who do not have the condition, it's gone from within that particular branch of that family tree. It results in anyone who's not affected having to be homozygous recessive. That is the only combination of alleles you can have if you do not have the condition. But people who are affected can either be homozygous dominant or heterozygous. Okay? They are the two options you can be if you have the condition. So again, if you're filling out a pedigree chart with all the genotypes, always start with the non-affected people because you know they'll be lowercase, and that's the way to work out an autosomal dominant one. Now, again, let's have a look at a wee example here from a past paper. We've got the inheritance of familial hypercholesterolemia, or FH for short, within 3 generations of a family. FH is caused by an autosomal dominant allele. How many individuals in this family are homozygous dominant for this condition? Okay, well, the hint I just gave you was if they're unaffected and it's a dominant condition, they'll have 2 lowercase letters, okay? So let's use an H for this one, okay? So 2 lowercase for all the people that are not affected, okay? So that's going to be lowercase, 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 lowercase. Unaffected, the clear boxes, everyone who's affected must have at least one capital letter, okay? So we'll put in that one capital letter for now. Because it's a dominant condition, you have to have a dominant allele in order to have the condition. But if we look at each of these people's parents or offspring, what we can quickly see is, for example, this person has offspring that has small h. That means they must have a small h to give to that offspring. This person here has offspring which are small h as well. So they must have small h as their second allele. And this person here has a parent who's got small h, which means they must have inherited a small h from this parent. So therefore, their genotype is going to be that. So how many individuals are homozygous dominant? Well, it's none. Okay, there's no individual that's got 2 capital H's. So the answer is going to be A. And as I always say to my class, I feel that's a bit of a tricky, sneaky one, but that's how you work out that one. Let's have a look at incomplete dominance next. So incomplete dominance is where there are 3 states essentially for the particular condition. So if we look at something like sickle cell anemia, that's a nice example. Sometimes they use thalassemia in the exam, but sickle cell is one of the ones they use. That's caused by an autosomal defective allele. Okay, so it is an autosome which is affected. However, because it's incomplete dominance, we don't have dominant and recessive alleles. Okay, what we have is alleles which code for the maximum expression of the phenotype. That's where we've got our 2 T's here. Okay, homozygous state produces a maximum expression. It's rarely expressed maximally, however. Okay, to get those 2 T's is very rare. In the heterozygous state, okay, that's the A and the T, and you can see there's no capitals or smalls there, produces a reduced expression of the phenotype. So you still have some symptoms, you just don't have the full expression of the particular condition. And then those who have the AA homozygous are not affected in this case, okay? So we've got the maximum expression, we've got a reduced expression, and we've got those who are not affected. So there's 3 outcomes for people due to incomplete dominance here. And you can see an example of a Punnett square there, okay, for incomplete dominance, where we've got a homozygous parent here, another homozygous parent here, and they produce heterozygote offspring, okay? So that means they'll have the reduced expression of the condition as a result. So let's have a wee look at an example. The example is thalassemia, which I mentioned a moment ago, and you can see the letters that they use are very similar in these questions, okay? So if you're unaffected, you have AA. If you're thalassemia trait, you have AT. And severe thalassemia, that's the maximum expression, that's going to be TT. So we've got an unaffected man, okay? Unaffected is AA. And a woman with thalassemia trait, so that's going to be AT, have a child. The chance that the child will also have the thalassemia trait, that's AT, is, well, we do our Punnett square, so capital A for one parent, and we've got capital A T for this parent here. We can see the 4 offspring are going to be A, and AT. Okay, so thalassemia trait is AT. How many of AT? 2 out of 4. 2 out of 4 is going to be 50%. Okay, so that's how we work out an incomplete dominance question. So the last type of inheritance we need to know about is sex-linked recessive single gene disorders, okay? And these ones are maybe the trickiest, I would say, to do out of all of them. Something like haemophilia is a good example of a sex-linked recessive allele, and it's caused by the sex-linked recessive allele, and it's more common in males and females, like all sex-linked conditions, okay? It affects many more males. It is not transmitted to a male from his affected father. And that's because when you have a sex-linked condition, males pass on their Y chromosomes to their sons. And sex-linked conditions exist on the X chromosome. Okay, so when you're passing on a Y chromosome, you can't pass on the condition. So fathers cannot pass on a sex-linked recessive gene, single gene disorder onto their sons. Okay, that's an important rule to remember. If a son has a sex-linked condition, they have inherited that from their mother. It needs an individual to be homozygous if they are female, okay? So this here is the genotype of a female who has a sex-linked condition. You see they need two recessive alleles, two lowercase h's there on their X chromosomes to get the condition. They'll be expressed in a male if they have one copy of the defective allele, and that's because it's expressed on the X chromosome only and not the Y. So therefore, because males only have one copy of an X chromosome, they only need one copy of a defective allele to have the condition, okay? And that's a good one to remember for males as well. They only need one copy. It also means that all daughters of affected fathers will either be carriers, so they'll have inherited this here from the dad. Okay, daughters get the X chromosome from their dad, sons get the Y chromosome. Or the daughters will be affected themselves if they inherit a recessive allele from mum and a recessive X chromosome from dad as well. Okay, so if we have a look at the pattern of inheritance in this family tree, we can see here we've got a parent with the condition, but then it doesn't appear in the next generation, but then it appears in the generation that follows. And that's one of the clues that tells us it's going to be recessive rather than dominant. And what gives us a clue that it's going to be sex linked is we've got a male here who's affected, we've got a male here who's affected, and we've got a male here who's affected. So because it's three males that are affected and no females, it's likely to be sex linked. That's one of the clues, okay? We can also see that when we've got an affected father here, that the son has not received that condition, okay? Because fathers can't pass the condition on to the son, but they can make their daughters carriers, okay? And that's obviously what the case has been here and here. By making the daughter a carrier, it's meant that the sons from those daughters have then managed to get the condition. So again, let's look at a couple of examples before we finish up here. We've got Duchenne muscular dystrophy. It's a sex-linked recessive condition, okay, sex-linked recessive, and it results in weakening of skeletal muscles over time. Okay, we don't really need to know that. An unaffected man, that's important to know, and a carrier woman have a child. The percentage chance that the child will have the condition is, right, so when we're writing out sex-linked inheritance, we do it by, first of all, doing an X and a Y for dad, for the man, and doing an X and an X for the woman, okay? Now, the unaffected man, okay, it's a recessive condition. So if you're a man who's unaffected, you're going to have a capital letter. So let's use D here for Duchenne, okay? So that means the male must have a capital letter because they are unaffected and it's a recessive condition. The woman is a carrier, which means that she has both a capital and a lowercase d, okay? Now, what we have to do is calculate the chance that the child will have muscular dystrophy. So we have to do a Punnett square. Now, we don't do anything different for sex-linked Punnett squares. We do exactly the same as we would for an autosomal condition. So we put our X, D, and our Y there within our Punnett square. I'll just draw the lines for this one just to make it clearer for you to see where everything is, okay? We put our XD in there, X capital D, and that X small d goes in here. And then we simply put these together in a Punnett square. So we get X capital D and X capital D there. So that person's going to be unaffected. We then have X capital D here and X lowercase d here. So that person is unaffected, but they are a carrier. So a female carrier, female unaffected. Here we have a male who is unaffected. We know they're a male because it's XY, and capital D, because it's a recessive condition, is unaffected. And then we have a male who's going to have an X and a lowercase d and a Y. So therefore, that male is going to be affected, okay? So 1 of the 4 offspring are going to be affected, which is a 25% chance. Okay, and let's do this last one for good luck. This one says the diagram shows the inheritance of haemophilia, a blood clotting disorder, in 3 generations of a family. Haemophilia is caused by a sex-linked recessive allele. Okay, individuals P and Q are expecting a third child. What is the chance the child will have haemophilia? So the first thing I'm going to do here is I'm going to say, right, sex-linked recessive, okay? So this male that's affected is going to have a recessive allele, okay? We're going to do the 2 X's for mum there. We're going to do the XY for this male here, for P, and do the XX for this Q here, okay? And then we'll start working out what their alleles are going to be. Now, we've got 3 unaffected offspring here, okay? Now, we know that mum here is not affected because it says they're unaffected, okay? And we can also see that P here, who is unaffected, is going to have a capital letter like that. So is Q for this first X. Now, we know that the dad has to pass on his X chromosome to his daughter, which means that this person here is going to become that. So X capital H, X lowercase h, X capital H, Y, okay? So therefore, we have to work out what the chance of their child having haemophilia will be. So we do a Punnett square for these P and Q individuals. So we've got an X capital H and Y for the male, and we've got a X capital H for the female parent and X lowercase h there. And we pop them into our Punnett square like so. So we get X capital H, X capital H there. We get X capital H, X lowercase h. Okay, so we've got a carrier. Here we've got X capital H, Y. So that's an unaffected male, and X lowercase h, Y. So we've got an affected male. Okay, so it's similar to the one we looked at before. What is the percentage chance that this child will have haemophilia? Well, it's 1 in 4. Okay, so again, it's going to be 25% for that one. Okay, folks, I hope that video's helped and you understand topic 4 a little bit better. And I'll see you all in the next one. Class dismissed.